Yellow, red, and green, right? How are we doing so far? Yeah. We're trying to be COVID safe, girl. Okay. The next one is CCA. So, yes, green, green, red, right? So getting some greens, green, green, and red, right? And so the next one is TAT, so that's yellow, right? Red, and yellow. How are we doing? Can we do this by ourselves, or you guys still need me? Okay. I don't want to overteach here. Guys, I'm going to make a change here. Can you guys look up real quick? I'm so sorry, but I'm going to make a change here. Can you guys change that to a C? Can you guys do that for me? So if everybody could change that second to last space to a C, that would be great. Once you change that second to last space to a C, can you change on the coding to a G? Don't forget to do that. Okay. Did everybody hear that? Okay. Okay, so this is what you should have so far. How are we doing? My light makes certain colors look weird. So I've got yellow, red, and green, right? And then green, green, red. Yeah, you gotta have 15. Just add another bond. So far, so good, y'all? Thumbs up? Okay, let's go ahead and make the coding string. So if this is a T, then this has to be a what? A. And we know A is red. Huh? It's not backwards. What do you mean? Like, it's just a match. T with A with T, right? And so here's my DNA. Once you've built your DNA molecule, take a picture. And your picture goes right here. When you're done, take your picture. Try to squeeze them together as best you can. I know, it's marshmallow, so there's only so much squeezing you can do, right? But there's my DNA molecule. Yeah, try to put them together as best you can. You don't have to bond them. Just try to squeeze them together as best you can. All right, we good? Thumbs up? Yes, ma'am. How to insert a picture on the slide? So what you should do, what I would do is email my pictures to myself and then save the picture and put it onto the slide. Now there's other ways, other students have other ways of doing it, but that's how I would do it. You don't have to put the pictures on right now. You might want to just take your pictures in order and make sure you put them in that same order. Okay. All right, next up. Next up, we're going to open up DNA. So heads up. We're going to open up DNA. So open up your DNA. So unzip, unwind, like your RNA polymerase. And now we're going to make what? RNA polymerase makes what? What does RNA polymerase produce? Uh-oh. mRNA. So we're going to make some mRNA. What is this process called when we make mRNA from DNA?
Transcription.
Nope. Transcription. So let's go ahead and make some mRNA. Your mRNA is red. Now we've got some changes here because there is no T in RNA, correct?
Yeah, so it's U.
So it's U. Your U's are white marshmallows. So let's start building our mRNA. So right here, I've got a T over here, so I'm going to put an A over there on the mRNA side. But I have an A over here, so what am I going to put over here? For uracil. Good, and put a U. I have a C right here, so I'm going to put a what? Orange for G. And then let's go ahead and keep going. How we doing, guys? Are we good? No? Okay. How are you doing back there? How are you guys back there? You okay? You guys, okay. Dayton, how you doing back there? All right, so we're going to take a picture of our mRNA. Everybody's mRNA built. Is anyone behind? Speak up now, because I don't want you to be behind. Are we good back there? Thumbs up? Can I get thumbs? All right. So let's take a picture of we're going to close up DNA because we're done with it. Oh, poor DNA. Feels used, but that's okay. And we're just going to take a picture of our mRNA exiting the scene. So take a picture of your mRNA. And then that picture goes on slide 6. All right, the next picture is you putting your mRNA on your ribosome. Okay, so let's get our mRNA. Heads up. We're going to put our mRNA, and we're not going to put it on the E site. We're going to start on the P site. So there's your next picture. Take that picture. No, these two are different slides. So here's the one that was mRNA by itself was this slide right here. Are you looking?
Yeah.
And then the one where you put it on the ribosome, it is this slide right here.
Okay.
So this picture right here is slide 8. So this right here, the mRNA attaching to the ribosome, is slide 8. Are we clear? How are we doing? Okay. All right, so now that our ribosome is nice, our mRNA is attached to our ribosome, now we have to start making our tRNAs, right? So grab your big marshmallows. And like I said, if you don't have the 3 little toothpicks sticking out at the bottom, go grab some more toothpicks. We have plenty of toothpicks. Feel free to grab some more toothpicks if you need them. Now you need to figure out what the anticodon is to this codon. So I have A, U, G. What's the anticodon on my tRNA? What's the anticodon to AUG? Uh-oh. What's the anticodon to AUG? You don't know? So what would be opposite of A? What would you put? U? A? This is a G, so C. UAC. Okay, so let's put UAC on there. U, and then A, and we know C is green, so all right, here comes the tricky part. Now we have to put an amino acid on top, but we have to figure out which amino acid to put up there, right? So now we have to take out our codon chart, so please grab your codon chart. Grab your codon chart. All right, question. This is the codon, right? Yes? The mRNA has the codon. Heads up. Guys, you can put your pictures on anytime. Okay, so the mRNA has the codon, and the tRNA has the what? What's the opposite of the codon? The anticodon. So codon, anticodon. You with me? All right. So if this is the codon and this is a codon chart, which three letters are we going to use? The codon or the anticodon? The codon. We're going to use the codon. Okay. And so the codon is what? A, U, G. Using our codon chart, we have A, U, G. Which amino acid are we going to put on top? Uh-oh. Meth. We're going to put meth. That's good. So methionine is what it's called, but you can call it meth. So methionine is what color? Red. So we're going to put the red one on top. Ooh, a marshmallow does not want to stick. All right. How we doing? Bethany, you good? You know what we're doing, Vic? You good? Okay. Just let me know when you guys are confused. All right, so making a mess. All right.
Oh, I'm still trying to put them on. I'm just looking at them from my phone.
Here's our mRNA. Yes. The first codon was AUG. Yes. We found the tRNA that had the what? What is that at the bottom? Anticodon. We use our codon chart. So we use the codon to figure out the amino acid that's on top of the tRNA. Now I know in your mind you're like, Ms. Johnson, why can't we just use the anticodon? It's on the tRNA, we just don't. Okay, so we're going to use the codon that's on the mRNA to figure out the amino acid. Take this picture. That's your next slide picture. I'm not sure which number that is, probably number 9. Say that one more time? Yeah, it's just mRNA. Oh, no, this is slide 8. Sorry. This is slide 8 right here. This picture right here is slide 8. Going on to slide 9. All right, so now we get another tRNA molecule, yes, that comes along. And we've got to figure out the anticodon that goes down here. So the codon looks like GGU. So what's the anticodon to GGU? CCA. CCA. But we're not done because we need to figure out what the amino acid that goes up here could be. So what are we going to use? Are we going to use the codon or the anticodon? We're going to use the codon. So we know the codon is GGU, right? So let's get our codon chart out. GGU. Or we could use this one. GGU. Is anyone still confused about how to use the codon chart? Make sure you let me know. So GGU, which amino acid are we using today? Which amino acid is it? Glycine. And glycine is the color orange. So I'm going to grab an orange. Okay? This is what it should look like. Please take your picture. This picture goes on slide 9. How are we doing so far? Okay, that's one group. Allison, how are you doing? Okay, Dayton? I can't see you guys from over here. Caden, how y'all doing? Good. All right. Kaylin, life is good? All right. All right, moving on. So now we've got 2 tRNA molecules, right? The next step is that we're going to bind these two here, these 2 amino acids together. So this amino acid is going to pop off and bind to the second amino acid. Okay? So this amino acid pops off, and like the traitor it is going to bind to the second amino acid. Say it again. I can't see. You can come. Bring it here. Sorry, I can't see through the screen. I'm sure it's fine. Okay, once you've done that, here's the new process. Now we're going to shift, and this tRNA is no longer necessary. I know that seems mean, but we're going to let that tRNA go, and we're going to take a picture. That's slide 10. Take a picture. That's your slide 10 picture. It looks like that. So that tRNA is no longer useful, right, because it doesn't have an amino acid anymore. So we're going to let it go. And what is that bond called between the two amino acids again? Peptide. Good. Can you guess what's next? What the next step is? Another, what is it called? All right, so after you've taken that picture, did you guys take this picture? Thumbs up. I don't want to go too fast. So after you've taken that picture, now we're going to do it again. So now my codon, it says A, U, A. So what's my anticodon? What's the anticodon to AUA? No, what's the anticodon? What goes up here? A binds with U. Very good. All right, now I've got to figure out what amino acid goes on top. But bear in mind, I cannot use the tRNA anticodons, right? I have to use the mRNA codons. I have to use these. So it's A, U, A. So I'm going to grab my codon chart. A, U, A. What amino acid does that code for? It's isoleucine. Don't you wish that the words made better sense? So isoleucine is what color? It is green. So I'm going to put the green dot on top. All right, and here is your picture for slide 11. What is the next step in the process here? Exactly. So I'm going to bind this one to this one. I don't need that first tRNA anymore. Bye-bye. It's been real. I have fun. It feels used. That's okay. And I'm going to shift. Did everybody notice that I shift? I'm sorry, I didn't show it to you, but I shifted over to the left. Right? Did you guys see that? Yeah. Shifted over to the left. This tRNA can start leaving, and then this is your picture. This is slide 12. Your slide 12 picture. This is slide 12, yes. Can we guess what happens next? What is the next step in the process? Another tRNA comes along. This seems a bit repetitive, doesn't it? So long as you understand the process. I know it's repetitive. Okay, I need to figure out what the anticodon is. So I've got, looks like CCA. What would the anticodon be? GGU. Okay, now that I figured out my anticodon, I put my anticodon on my tRNA. What amino acid goes on top of that tRNA? Dang, girl. All right, proline. Okay, let me show you guys how she figured that out. So I've got what is this? CCA, right? CCA, proline, and we know from our first slide that proline is, it says proline is yellow. So that's how she figured that out, if you guys didn't know. So I'm going to put proline up here. Okay, and then take my picture. Take this picture. Thumbs up when you're ready. Okay, good. Yos is ready. You guys are ready? All right. We're good? What's the next step in the process? What do I got to do? Okay, I need to bind the amino acids. This one's going to pop off. We don't need it anymore. Now it's going to float away. And we're going to shift. And take our picture. Notice you should be out of tRNAs by now. Did you guys notice that? Which ones are your tRNAs?
Oh, the big one.
The big marshmallows are your tRNAs. So do you notice that you're out of tRNAs?
Yeah, we've been out for a while.
Mm-hmm. You've been out for a while.
Yeah, I've been recycling the skin.
Really? So the people before you just ate the tRNAs?
I heard you really.
That's special. That's special. All right. So you should have had 4 tRNAs, but okay. Anyways, so let's look at this last codon. I have U, G, A. Did you guys notice that? All right. Now, take out your codon chart and tell me what UGA codes for. Guess what we're going to do? We're going to stop. Okay? So UGA codes for stop. So we are done. Did you guys already post your picture for slide 14?
I'm not working.
What's not working?
Like, I'll try to send one to my school email and then it's all like white.
Just-
yeah, because it blocks the pictures from that because it's like, it could be like,
What?
I know, so it only lets like y'all, like, teachers send it. So like, people like in the school can't send it to each other.
That's ridiculous. Okay, that's okay. We'll figure something else out. That's stupid, but we'll figure it out.
Okay, let me do two of them. Now it won't let me send other ones.
And that also might be internet.
Yeah, it could be this.
All right, so this right here should be your slide 14. This picture right here, right? Are we good?
Yeah.
All right, so here's slide 15. Is just your polypeptide, your amino acid strand. So I'm going to take this off. So it's just this, the amino acid strand that you just made. It's just this. So I've got amino acids. The bonds are called what? What are these bonds called?
Oh,
Peptide bonds. The whole thing is called a polypeptide, or AKA protein. That goes on slide 15. No, slide 15. Good time. Take all of that off now. Good times, yeah. Just take the marshmallows off. That's it. Oh, and the dots. Take the dots off. Just the marshmallows and the dots, but the bonds you can keep. All right, so the biggest part of this is not so much your pictures, is this last slide, slide 16, and it's your conclusion, your summary. So you're just going to summarize this whole process. Now, if you're still confused about the process, like I told the folks that are at home, there's a video that I posted, so you can watch the video. You know what I mean? So you'll be like, oh, this video helps. Thanks, Miss Johnson. You can use that video to help you write your conclusion. How about that? And then this is not going to be due until next class, okay? So what I need for you guys to do is go ahead and head on back. To your desks. We're going to go over mutations. All right, can you guys hear me at home?
Yes, ma'am.
All right, cool. Okay, so we're going to talk about mutations, and these notes I'm going to write. So what I'm going to do is I'm going to record it for you guys, but I'm also going to post a picture of my notes so you can copy it. And if you get confused, you can go back and watch the video. The problem is that I can't write and display for you guys and for the students in class at the same time. I just don't have the technology. So that is going to be the plan. So let me post this picture for you guys. Yes? Absolutely. Nope, that's not what I meant to do. There we go. And then here's the second one. Okay, those of you at home, so Tiffany and Sarah E., if you guys could look at the chat real quick. Those are the notes that we're about to do. Okay?
Already.
Okay. I'm assuming you can't see this, right? You can't see me writing? Can't see my hand?
No, ma'am.
Okay. But I am recording, like I said, and those are the notes. All right, guys. Let's talk about mutation. What is a mutation? Can somebody kind of give me an idea of what they think a mutation might be? Excellent. That's good. Okay. So when I say that we are mutating DNA, I mean that we are changing that DNA. We're changing DNA. So that's what mutation is a change in DNA. I have notoriously bad handwriting, so you guys just have to make sure that, you know, if I'm writing terribly, that you have to just kind of let me know. Okay, so mutation is a change in DNA. So if we change the DNA, we know that DNA gets coded into mRNA, and then mRNA is used to build protein, right? So if we change the DNA, we change the mRNA as well. Does that make sense? So a change in DNA means a change in mRNA, which could mean, not necessarily, but could mean a change in the protein. Now, if we change that protein, then we could end up changing whatever trait it codes for. So, for instance, if we talk about sickle cell anemia, does anybody have family or know anyone with sickle cell?
Yeah.
Okay. So sickle cell anemia is actually just one base change in DNA. Literally, it's just one. And that one base change causes the protein hemoglobin to be misshapen, and that misshapen hemoglobin doesn't carry oxygen very well. And so those people are not able to transfer or transport oxygen very well in their bodies. And that could be a very big problem, obviously, right? Not the ability to not get oxygen from your toes to your head and back. It's a major issue. So, may change the trait. All right, so that's our little background real quick. And then, so there are two main types of mutations. And as always with biology, there are multiple names for stuff. So two types of mutations. Okay, so the first type of mutation is called a point mutation. I'm going to put that over here. So point mutation. And then the second type is called a frame shift. So frame shift. Now, sometimes a point mutation is called a substitution. Okay, so sometimes it's called a substitution, but frame shift's always called a frame shift. Okay, so starting on the point mutation side. So a point mutation is a change to one letter or one base. Okay, so remember all the letters are nitrogen bases, right? This might change the protein, or it might not. Okay, so it might change the protein, it might not. Okay, we have three types of point mutations. The first one is called a missense. Okay, a missense is when we change one base or one letter, and that changes the amino acid. So let me give you an example. So here's the correct amino acid, I'm sorry, the correct mRNA strand. So if I have AUG, and then CGU, and then CUA, and then UAC, and then GCA, and then the last one is UGC. Now I take out my amino acid, my codon chart, and when I take out my codon chart, what amino acid do I code for if it's AUG? AUG. So AUG. The amino acid is methionine, right? And then CGU. CGU is what? Arginine. And then CUA. CUA is leucine. UAC. Tyrosine. And then GCA. Alanine. And then UGC. cysteine. All right, so that's our normal, right? That's the normal and correct amino acid strand. Now let's write the mutated one. In other words, let's change it. Let's mutate it. Instead of having a C right here, I'm going to change it to a U. Okay, so everything else is the same. AUG, CGU, CUA, UAC. Now instead of GCA, it's going to be G. I'm going to use a different color. It's going to be G, U, U, A. So everybody see that change? And then U, G, C. Does everybody see that change? That's a mutation. How are we doing so far? All right, so can we all agree that the amino acid strand will be the same here? So MET would be the same, right? Arginine, leucine, tyrosine, all of that would be the same, but will we still have alanine right here? No. So let's look at that. GUA is going to be what? GUA is what? Come on, guys.
So what was it? Valine?
Yes. So valine. So now I've got, bless you. Instead of alanine, now I have valine. Okay? And then cysteine is still the same. Does everybody see the change there? So you're probably saying to yourself, but Ms. Johnson, it's just one amino acid. What difference does that make? It makes a big difference. This is the correct hemoglobin. This is the incorrect hemoglobin. This keeps you alive. This could kill you. One base change. Does that kind of make sense? Jayla, how you doing? You good? You guys just let me know when I'm losing you. All right, so that's a missense. So like I said, the example there is sickle cell. All right, so another one is called the silent mutation. That's when the base changes, but the amino acid doesn't. So that's a silent mutation. So nothing happens. So let me give you an example. So here's your correct strand, same one, AUG, CGU, CUA, UAC, GCA, UGC. Right? So that is your correct strand. And so the amino acids are methionine, arginine, leucine, tyrosine, alanine, cysteine. So that's correct. Now let's mutate it. Okay, so I'm going to change that GCA to a GCU. So AUG, CGU, CUA, UAC, GCU. So that's going to be the change right there. That U is the change right there. GCU, UGC. So all this is normal. And then we get to GCU. Using our codon chart, what is the amino acid that GCU codes for? Alanine. Well, wait a minute. Isn't that the same? So is there any change to the protein? Will the protein change at all? No. That's why it's called a silent mutation. There is no change. Okay? All right, we're going to have to flip over to the back because I ran out of space. There's a third point mutation. So point mutation number 3 is called a nonsense. A nonsense mutation is when we change to stop. So when we stop it in the middle. So we're going to change to stop. So let's write that one. So here's the correct. It said AUG, CGU, GUA, UAC, GCA, UGC. That's the correct. And then so the amino acids are methionine. And then what was the second one? Did arginine, leucine, tyrosine, alanine, and cysteine. So that was the correct strand, right? Now let's mutate it. Let's change it. Let's change it to, so I have AUG, and then CGU, those are still normal. GUA, still normal. And I'm going to change this C to an A. So UA, I'm going to change this to an A. Does everybody see where I changed that? So change that C to an A. All right, and then I got GCA and UGC. Okay, now question to you. I know that this one's going to be methionine, like normal, arginine, like normal, leucine, like normal. But what is UAA? What does that code to? It codes to stop. So guess what we're going to do? We're going to stop. Is that protein finished? No. Do you see how that could be a problem? That means that that protein is halfway done. And it's just one base change. You see how it's just one base change can change the whole thing? So 3 types of point mutations. Missense changes the amino acid. Silent doesn't change anything. And nonsense causes it to stop in the middle. Now, the other type of mutation, flip back over. And this is the end, I promise, is the frame shift. With the frame shift, we're going to do an addition or deletion of bases. So we're going to add or delete bases. Okay, so obviously that always changes the protein. Okay, it always changes the protein. So the first type is called the addition. Guess what that does? What do you think we're going to do with addition? Add a base. All right, so here's your correct. And again, we're just going to use the same one we've been using. AUG, CGU, GUA, UAC, GCA, and the last one was UGC. We know that that is methionine, arginine, leucine, tyrosine, alanine, and cysteine. How are we doing, guys? Are we good? Now let's mutate it. Let's go ahead and add a U. So AUG, CGU, GUA, UAC, and I'm going to add a U here, so I'm going to put GU. I just added a U right there. I just added a U right there. That's where I added it. And then I'm going to continue. So I'm going to put the C, and then AUG, and then I'm going to have a random C right there. Let's figure out how that changes everything. So I had AUG, that codes for methionine, arginine, leucine, all that's normal. But then I get to GUC. What does that code for? Does it code for valine? GUC. Okay. Are we good? Does everybody know how to use the code? Jayla, do you know how to use your codon chart? Okay, just making sure. All right, so this codes for valine. And then AUG codes for meth. Good. Does everybody see how that changed it? All right, cool. Last one. The last one is called a deletion. Guess what we're going to do with the deletion? Delete one. So we're going to lose one or more bases. All right, again with the correct strand. Oh my gosh, I think I have this memorized. AUG, CGU, GUA, UAC, GCA, and then UGC. That's the correct. And so we know that the amino acid strand, the correct amino acid strand is methionine, arginine, leucine, tyrosine, alanine, and cysteine. Now let's mutate it. So we said we're going to delete one, right? All right, so I'm going to go ahead and delete. I'm going to delete this C right here. I'm going to delete that C. So let's delete that C. So AUG, GCU, GUA, UAC, but I deleted this C, right? So now it's going to be what? GAU, right? Because I deleted the C. Does that make sense? And then I'm going to have this weird, like, GC at the end, right? Does that kind of make sense? You guys are super quiet. Let me know. Now, once I translate it, that first amino acid's right, second one's right, third is good, tyrosine is good, but then I get here, what amino acid does GAU code for? Say it again. Very good, aspartic acid. So GAU is aspartic acid, right? So I'm just going to put ASP there. That being said, can I get another amino acid right there? It's not complete. Do we see how that's changed? All right. Your example disease for this one is cystic fibrosis. Does anybody know anyone with cystic fibrosis? This is the exact mutation that occurs. What happens is there's a sodium-potassium pump. We talked about those. Remember sodium-potassium pump? This particular code codes for the sodium-potassium pump in their lung cells. And since it's messed up, the sodium-potassium pump is messed up, and it does not pump out the ions. And what happens is they develop a nasty little kind of a mucus lining, a heavy mucus lining, and so it's kind of like breathing, like having bronchitis all the time for them. They don't live past like age 32, so they don't live very long. All right, those are the mutations that'll be on your test, okay? Do we understand that? You're going to have to know the difference between the point and the frame shift. Please turn in your notes. These should be the last note submissions that you turn in, is the mutations. And then your homework will be the conclusion for your lab, as well as finishing